close the system had to be to my brain to feel like it's me, to be the same guy. That's then there's the question of given a certain amount of behavioral data, physiological data, then how closely can you reconstruct? How well can you solve the inverse problem? We have no idea of the answer to either of those two questions that I just named. And solving either of these two questions is quite hard, right? So I mean, it's a possible solution, which has a non-trivial chance of working. To estimate the odds of it working requires two breakthrough discoveries, which may or may not be harder than building an AGI or anything.
Right, so that was the Bainbridge-Rothblatt me program idea again, but there's really no other notion.
I already have kids.
But a more approximate representation.
Well, that's a cool thing to do. It's not awkward. Well, making another guy sort of like you.
It's a good response.
Someone made a comment in the chat I thought was right on. It said, first while and just press down, we'll end up with an expert that someone could have used that. Or tell him what his dad might have said or done.
Maybe he'll like that.
He might always say that. Oh, yeah,
but there's also certain value to just capturing information, right? I mean, we value with photographs from the 19th century because it gives us a vision of what happened in the past. I mean, why do we value literature? All the literature and history of the ages, the readings of scientific data represent some kind of capturing of past states. So, and those fuel future theories. You can refer back to that data. So the question would be, with like capturing a portrait, maybe a sort of a living algorithmic portrait, even if it's not an accurate portrait of a person, you're not making the person live. In other words, it's not the full me. It's part of me. It's a memory of me. So it's like these sort of higher, decent, high density dimensional portraits of who we are as individuals. We get value from like Renaissance paintings for that same reason, you know. Why else is this camera on our phone if it's not because we intuitively value that storage of the information? So can that sketchy approach, the sketches of who we are, then help to inform cognitive science, neuroscience, and then maybe iterative over time a set of tool, a huge corpus of data for helping to address these deeper questions.
So if we describe
Yeah. Okay. So this is interesting because what you're saying, basically what several of you have been saying, which is that you could use something similar to a Rothblatt method, capturing data to create something similar to yourself, goes back to the previous question of, does what you're trying to do with these mind functions you're recreating, does that impact the resolution you need? Because if all you're interested in is something that is similar to yourself in some way, then perhaps what you need to capture in terms of data is much easier to gather. But before we go on, or before I ask Ramez what he wants to say, I just wanted to throw something else out there, which is that if we think in terms of how to simplify the process of creating in an individual person a real upload, let's just say, There are, yes, this inheritance, multiple inheritance problem. I like thinking of it in that sense because we can, of course, consider some things that are in every person's brain that are the same. All our brains are not completely different. There are large pathways that are the same. There are regions that do the same sort of thing. So maybe we can't use that to create a me program because maybe the delta is just as big, has just as much of an amplitude as the average itself does, or as, I mean, average is the wrong word for that then, but as the generic version would have, so that you would have to look at just as much data. But what you could do with this generic version is you could validate. So you could at least test, perhaps, if you're making any big mistakes, if you've left out an important pathway that somehow didn't show up on your scan or something like that. Let's say you use the Atlam to scan the brain and get the structure, but something didn't show up in that one individual brain, but you do know what brains are supposed to look like. You've got a generic version. And you can use that to correct problems, validate. I'm just throwing that out there. That's potentially a way to solve some problems.
What if the construct that you're making isn't the exact view? You're connected to it after you look at the data, and you're transmitting data to it from it. So it's kind of a living extension of it. So you're running the copy in some data center somewhere, right? And it's as close as you could get from the therapy specifics you added to it. Now you're in constant communication with it wirelessly. Okay, so that you can extend yourself into it, okay, and you now have effectively kind of think of it as an extension of you.
Right, so a gradual uploading or offloading based on augmentation and augmentation, neuroprosthetics, BCI, some kind of extension like that. That is one of the things what brought up as, say, one example of a potential other direction to go, or something that will naturally evolve as everything in the context of what we were discussing about is progressing. That was yesterday. We came up with that stuff as well. Yeah. Ben? Yes.
I was thinking, I'm just brainstorming a bit. I mean, if something along the lines of Martine's idea was going to work, you can imagine, like, if you had some mediocre method of scanning, non-invasive method of scanning the brain, like some little cat you work all day as you go around doing everything. It's kind of crudely gathering information on your brain. Maybe constant real-time crude data over years give you the same thing as a really good scanning method over a short period of time. Then you can all piece that together. That would seem to be more hopeful than getting it from my diary videos, getting speeches.
I have about 2 comments. So one, the last one, my guess is it's very challenging to find that method of data collection for with a structural system. It's very difficult to take the composite personality traits that you found and actually plant them in relations that works well with the brain. If you did, you would have a group that would, on introspection, start to discover odd things about itself. On the other child memories, it would probably believe that it belonged to something somewhere else, which told them exactly what it's called tapped. So that organism is not history, personal narrative, something that actually getting back to your question about is there a simpler way to reduce the data? One thing I was out there is stuff that the idea is doing is instead of scanning actual mice brains, some idea of what the technology of mice brain looks like, some average model based on that, could potentially get into something that's a generic but not. There are some challenges with that, in terms of pointing out a function of the brain or contents of the brain, if you will. And like the modem, whatever you call that, it's like mice who obey at some point where the brain very
Yeah, it would be a construct, but could you graft a specific mouse's experiences into this general construct?
That requires kind of do the translation from experience to neural circuitry. And that by itself is Wednesday part of the problem. I'm not sure how much we're actually getting.
I kind of disagree because, like, if you're going to model with an adult mouse brain, then it would have gone through gestation, it would have interacted with mice, and if you were going to generalize, then what you would get would be general experiences. So for human beings, you know, we all experience certain aspects of the world that are kind of similar, right? I mean,
It's going to be we have like implicit abilities that some averaged out set of neurons
universal brain.
where it's for a man take the whole of the computer like everything. And
You have no idea.
Where here's a new idea. I'm going to have a little bit of sponsored by her. Like your question. If you imagine the model the development process. So imagine that what you used was something that went through that model and where the brain was at just pre-birth and gone through sort of average gestational process, and then could emerge instead of actually at an average, and then we go through a learning process. That might save them a bit of motion.
Right.
You're not going to have an AI that's going
Well,
to program that AI.
it is entirely possible, of course, that if you wanted to create some generic brain to use as your template that you then try to graft specific characteristics in, that you would not use an actual average of all the data, but you would instead use a model that would work. So this model could be developed through the method you just described, basically from gestation onwards, or you could just use, you know, neuroscience expertise to try to put together a model that works. So you could try to do that. And then the question is, what do you do with that? Can you put individual characteristics into that? Can you somehow just modify it so it becomes an individual brain? Or do you just use it for verification purposes or something like that? I mean, what can you do with that? That's really the point then. But we should really, this should be the last bit on this topic because Peter really should get the chance to finally give his talk since he didn't manage to yesterday because of our rescheduling, and now he really has to get a chance. So, Ben.
Feels like, just thinking in terms of my own kind of AGI system as well, introspective, like mapping, somehow imprinting a known experience into this mind is a highly strained and non-deterministic process. Like imagine taking myself, so imagine there was some new experience. Like, okay, what would I be if I had once run naked in the streets of Calcutta, right? Then I graph that in. That can go a lot of directions. I could have had many random impacts on me. How can I say what, right? And the same in an open con system. You can decline some memory 5 times, I get a different result each time. So you can't just incrementally graft into memory. It's like some global optimization process of given this vast amount of things, what's the most resilient way to put them all together? Fantastically hard optimization problem. It might have very many local optima, which might be actually different. It's not at all clear that you're going to get you 50% of your experience with the optimization. Although you'll get a bunch of guys interested in the background.
Right. So we can never do that experiment you see in the cartoons where you have the bunny and the chicken and you just switch their brains by, I don't know, grafting their memories over or something. Anyway, okay. So let's move on to Peter's talk. You're up next, Peter. Yes, it's tapped. Yeah, the screen blanks away, yeah. Slides are in the room. I don't know the exact title right now, but he's talking about a functional approach to brain emulation, basically. Okay, there he goes. Is that him or is that you? That's you. But I guess if you can get the slides up. Slides are in the room, he says.
Hey guys. I'm over here behind the lecture theater. My slides are on one of the boards right there. I just need to move them to the lectern.
Okay. There we go. Uploading the stored object. We're going to put it on the lectern.
I love it.
There comes Peter. He is on stage.
Okay, I'm going to turn on the webcam now. You guys picking me up okay?
Sorry. Yeah, we see you. I mean, I see you, but we don't see you on the big screen. Oh, we see you here. Okay, we've got you on the other monitor. Yeah, you're visible. And
Okay.
we don't have your slides up yet, though.
They were just on the lectern, and then they disappeared.
Oh, there is Julio. Okay. Yeah. No, the only thing that I've done ever is to just take the same file and put it on the lectern again, but then I had the file locally.
I'm looking for Peter's slides. I have copied them to the inventory. What was the name of the slides, Peter?
Okay, Julio, she's got it. Suzanne
Okay.
has it. Thanks. There we go, document going in there. Oh, you put the display panel in. Okay. I see. There we are. The
Okay.
presentation is there. Perfect. Okay, Peter, anytime you're ready.
Okay, just had a question about how I interact with the room. Should I just go through this whole talk, or will you guys stop me if there's questions in the room?
If there is a question in the room, then I will convey it to you, okay?
Okay, sounds good.
Okay.
All right. So what I wanted to show you today is give you a little overview of the project I've been working on for nearly 5 years now. And so earlier, the idea of using a nematode and doing a full model of the nematode brain, because, well, nematodes don't really have brains. They have a distributed nervous system with several ganglia. But so I went through looking at a bunch of different model systems, and the one I selected to use.
Words like uploading, because that's very confusing, but it's the same basic principle. The idea is take the functions of the mind.
Stop that recorded session. Thank you. All right. So the model system I selected is a mollusk, is a snail brain. And so I'm going to give you a quick overview of my perspective on whole brain emulation, how we get there, and the testbed system that I've been using for the last 5 years. Let me go to the slides here. All right, so these are the questions that I think about for whole brain emulation. So the first thing for me is that we need to identify what is the information we actually require to do this. And this is the resolution and scale question again. So answering this question, I think, requires that we work with testbed systems in order to develop some experience with how we parameterize the low-level variables to get to the higher functional dynamics that we're interested in. Along the way, we need to decide how we're going to acquire that information.
Peter, I think we just... Oh, we lost your camera. There's an exclamation mark next to his name, so it seems like he may be having some connection delay problems. Yeah. Well, obviously that was going to happen. There he is. Okay. Oh, he's in the lobby. I don't know, Peter, can you hear me while you're in the lobby? Oh, here he is. Okay, he's back.
Hey guys, Teleplace crashed on me. All right, can you hear me again?
I can hear you.
Okay, starting my video again. That's the first time that's happened. Okay, so where was I? All right, so how do we acquire that information? What type of sensors do we need to get the information we'd like to have? So again, questions of scale and resolution. If we can acquire that information, what type of frameworks do we need to understand that information? So in terms of software, in terms of intellectual models. So that's, basically how do we frame all this information that we've acquired? And then we want to actually implement this as a model, as a system that we can exist in as an alternative substrate. So how do we turn those frameworks into physical systems? So these 4 questions form for me the way to think about how to experimentally attack these problems we're talking about. All right, so what information do we need? So, go back one there. So the information that I'm focusing on right now are spikes, spike trains primarily, or voltage records, because the system I'm working on, this Snail, has quite a few electrical synapses as well as chemical synapses. So I need sometimes more than just spiking information. But I'm focusing on single neuron voltage records, and that's not because I think that is all I need. It's just the most accessible data right now. But I'll talk about that in a second. But there might be some other things that we need, particularly neuromodulator concentrations. So we need to look at these large-scale variables across the entire brain to see how they're affecting the dynamics across large areas of the brain. We might need to look at things like glial modulation. So glia are the support cells. Probably everybody in the room knows this. But there's not, particularly for mammalian systems, there's a lot of questions out there still as to how glia are actually modifying the dynamics of what's actually happening in the brain from moment to moment. We probably need large-scale structure. We need the connectome. We need cell types and locations, and we need an axodendritic map, so that the connectivity down at the level of axons and dendrites to really, if you really wanted to build up a model of an individual person, you might need all of this structural information in order to parameterize your model. And at the worst case, we might need to know the machinery of single cells. We might need to know down to ion channel signal transduction molecules, or even down to molecular states and things like that. We just don't know right now, but I think there's some reasons, some very good reasons for starting at the spiking neuron level. All right, so the reason to start with the spiking neuron models is the type of dynamical model you'd want to start with is because this is the densest, the voltage records and the spiking communication in between neurons is the densest information flow that we know of in biological systems currently. It's easily detectable with external sensors, and there's a well-developed analysis rationale for using spike trains to describe both sensory input and motor output. All right, so the system that I'm working with, the sensory system I'm working with, sensor system I'm working with is multi-electrode recording. The highest resolution that's currently available for this type of system are 4,096 electrodes over about 2.6 millimeters square. The system I'm currently using has 252 electrodes over an area of about 1.5 millimeters. I'll show you some examples of that in a second. Sampling rates, we need about 10 kilohertz, we think. These systems can go up to about 25, 30 kilohertz. We're using electrodes that are 10 to 30 microns in diameter, and there's several different array formats and electrode types in use. We're using what's called a planar multi-electrode array, which is a glass, essentially a glass petri dish with embedded electrodes at the bottom of it. So we're looking at surface level data. Let's talk about the Snail Testbed system real quick. So for me, this is a nearly ideal system from a single neuron analysis perspective. If you're going to build up a model of a simple brain, this level, so this type of mollusk is about as simple as you can go and still have the system be considered a true brain. And what I mean by that is that all the functions are organized in a way that generally resembles what we see in higher order systems. If you go below this, you don't have the same type of organization. So you really do have behavioral organization in particular centers in mollusk systems. So Limnea, the system that I work in, this aquatic snail, is a good example of that. The much more widely used Aplysia is also an excellent example of this type of behavioral organization. So what are your advantages for working in a system like this? You have large, so up to 150 micron in diameter cells. They're visually identifiable. They're fairly, very consistent from organism to organism. These cells tend to be in the same place and have roughly the same behavioral function in every organism. They're very identifiable input and output streams. So your sensory and motor streams are easily identifiable. You can go in and actually see the information coming into these systems, and you can see the information going back out of these systems. So it's very observable. The CNS has about 20,000 cells, so it's a manageable size in terms of the complexity and in terms of modeling. 60% of these are on the surface, as well as 60% of these cells are on the surface of the CNS, which is important for us because the recording technology we're using, we're limited to the surface. I'll talk about voltage-sensitive dyes at the end, which is one possible technique for getting at a more complete picture, maybe using that in combination with the technique that I'm using. And then the system has 11 well-defined ganglia that are organized into, this is sort of the simplest format of a brain that neuroscientists think of as a brain. Guys, you'll have to excuse me, it's 5 a.m. here, so I'm still a little sluggish. Okay, so this is the system I work in. This is the brain. This is a model of the Limnea brain, 11 ganglia. We focus on the feeding system right now as for really test bedding the ideas that we're working with to reverse engineer circuits. And so most of the feeding system is contained in those two small ganglia you see will be to, which direction? Well, the small ganglia to the right of the screen there, the buccal ganglia. So they contain all the motor neurons and part of the central pattern generator that generates the feeding behavior for the system. The other area we look at with some of the larger arrays we use are the cerebral ganglia, which is equivalent to would be like a cortex for the snail. Most of the sensory data passes through there and goes out to the other ganglia through that part of the system. Okay. So this is what it actually looks like on the multi-electrode array. The image to the left, you've got the buccal ganglia sitting on one of our sort of medium-sized arrays. The image to the top right, a ring of the buccal ganglia and the cerebral ganglia. So this is where we've just taken out part of the system and looked at the dynamics in that. One of the advantages for the system is it's very survivable outside the organism. So we now can literally take the entire brain out of the organism, leave sensory structures attached, and do experiments with it. So it's completely observable. Okay. This is one of the large high-density arrays we're using now. It's just 252 electrodes over 1.5 millimeters square, and that's the entire CNS of the organism with the lips and esophagus still attached. So we can do in vivo learning experiment, or ex vivo learning experiment. So with the brain out of the organism, it will live and be stable for about 2 to 3 hours, and we can do experiments such as providing it with a neutral taste and then immediately providing it with sucrose, and the brain will learn then to associate the neutral taste with being fed. So we can actually observe these changes going on in central nervous system when we do these types of experiments. Okay. So that's how we acquire the data, is with these multi-electrode arrays. From that, we get single voltage records. We get a lot of data back from this system. Experiments are now into the gigabytes for an hour or two experiment. And so the problem we're facing now is how to parse through tons of this data, and that's most of what I've been spending my time on for the last couple of years. So feature extraction, finding the important signals and sources in this data. So we want to do spike sorting. We want to locate individual cells. We want to know what they're up to on a moment-to-moment basis. We want to know where they are, roughly what size they are, and then we want to find the information connectivity between these sources, between these neurons. Who's talking to who in any particular instant, and then correlate that back with the behavior we see either ex vivo or in vivo. And then from there, if we can get that information, if we can get the individual cells and we can get the connectivity between them, building models, large-scale models of how these systems work and what happens, say, when you do a learning experiment, or what happens when the organism does a particular behavioral task. Excuse me. Okay. So this is sort of where we are right now. This is the type of results we're producing. So this is a 60 electrode array. Off to the right, you'll see there were 2 buccal ganglia sitting off to the right in that image, where you see the more dense and larger cells. And so we're able to get back these automated maps now, where we can run data through the analysis system that I'm working on. It will locate your signal sources and estimate the size of the cell that put those out. And then using a technique from a statistical connectivity analysis technique called Granger causality, we can start to look at the dynamic moment-to-moment connectivity of who's talking to who in these systems. So we build up these connectivity maps, and then we look at how these connectivity maps change during a learning experiment or during particular behaviors. Okay. So that's the quick version of what I'm actually up to. All right, so where do we want to go from this? And what are the lessons that we're learning from this? So sensor-to-neuron ratio is critical for good analysis. So if you can have one-to-one sensor-to-neuron ratio, or better than that, more than one sensor-to-neuron ratio, it really simplifies your analysis process. Whether it's actually possible to get to this in something on the scale of the human brain, we're probably talking about going into nanotechnology, into getting that type of sensor levels. But I think it might be reasonable to do much less than that. You might be able to do a 10-to-1 or 100-to-1 sensor-to-neuron ratio in order to pick up the data that you need, if you can produce analysis systems that can sort out the data so that you could separate multiple neurons out. So I'm very interested in combining data acquisition methods too. I was talking about at the beginning voltage-sensitive dyes. So this is a way of visually looking at the voltage changes in neurons. And there's some early indications that using a combination of multi-electrode array to get very detailed voltage records from the surface, in addition to using voltage-sensitive dyes to get deep information, you might be able to get a complete perspective in these small brains anyway of all of the single neuron voltage activity in a given brain. So I'm also very interested in using structural and functional data simultaneously. So using the approach that Ken talked about, I'm assuming, I wasn't here for that talk last night, about doing scanning at a very high resolution. I'm interested in how you go from that structural information to functional information. So if we have these deep structural scans, very high resolution structural scans, can you take those and use that to generate the parameters that will be the initial conditions for modeling a particular system? So, and I think the way that you do this is you do experiments back and forth using both structural and functional information simultaneously. So if I get a particular structure out of an organism, what does the functional behavior look like? So what you would have to do is run the functional experiment first, get a very detailed picture of sort of the dynamical system space that CNS exists in, and then once you're done, take it and do a very high resolution scan, and do that for a number of organisms until you build up a picture of how structure and function correlates. And you could do this at various resolutions, both structurally and functionally. Data analysis is absolutely the critical intellectual bottleneck. We can, in these systems, we can generate tons of data now on what's, we can do these ex vivo experiments for a very long period of time. We can record lots of data. We can run it through different perturbations to see the system in different states. But getting through the data to figure out what that actually means is definitely the hard problem we're facing right now. And I'm starting to do a lot of thinking about how we eventually apply this to vertebrate systems. I think the most useful thing about this is it's a very good test bed for thinking about the problems that we will face as we scale this up. But I think a lot of the data analysis techniques that we're already starting to use will be useful at the vertebrate level. And we do use them in our lab for looking at EEG data and fMRI data as well. So figuring out where how to actually apply the lessons that we're learning from snail data to vertebrate systems. Something I'm thinking about a lot right now. Okay, well, I'll finish up there and I'll open it up to discussion. And you guys.
Thank you, Peter. Thank you very much. Okay, there's already a question coming right up. I'll relay it to you.
Yes, I'm saying a bit more about the nature of the intellectual bottleneck around data analysis.
Could you say a little bit more about the intellectual bottleneck around the data analysis part?
Sure. Okay, so there's a couple different levels. There's spike sorting, which is a notoriously difficult problem for neuroscientists, especially when using, if your sensor to neuron ratio is pretty low. If you have 10 or more neurons coming in per electrode, you'll sometimes get a lot of you get signals that overlap between different neurons, and it can be very hard to separate them back out. So that's one of the really hard problems we're facing, is actually separating these neurons out in what we call feature space. So that's something I've been working on quite a bit. Then this whole area of functional connectivity analysis is really hot in different areas of neuroscience. It's hot in fMRI, it's in EEG. So say, for instance, if you're doing EEG, you want to look at the correlations between different brain areas. In the system that I work in, we're looking at actual neuron-to-neuron correlation to see who's talking to who at a particular time. So developing right statistical techniques for basically, if you have a bunch of different sensor streams, how do you figure out where the information is flowing from one piece of the system to another. That's one of the really big problems we're running into right now.
Thanks. Sorry, Sim has a, do we have a
Hi.
question from the virtual?
Hey, Sim.
Hi. Can you hear me in the room? Great. Just as a point of order, the conversation has been fascinating, and I'd just like to say that just as a point of order, can we just decide whether the people in Teleplace should be sort of included in the meeting? Because if we had the radio mic, this wouldn't be a problem, but it didn't work. So in order for us to hear the discussion, it would be good if people could come to the mic to make their point. Now, I realise that may stifle conversation, and people may be inhibited to speak, but I think maybe you could just have a collective decision on whether you would like to do that or not, because, and this would be the perfect moment to do that, because then you could talk directly to Peter. So maybe you could just sort of collectively decide whether you would like to include us or not. Thanks.
Thanks, Sim. All against?
Although I'm paying attention.
Okay, it seems, Sim, that that you have. Yeah, people are going to come up to the mic to ask questions. Good. So, yeah, Max.
How many people are in?
Oh, there. Gosh, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, like 13 or something.
Hey, so just a quick questions. One is the functional connectivity software that you showed, is that open source right now?
Not yet, because we haven't published it yet. But I'm desperately trying to get a paper out on it right now.
Okay.
Yep.
The other question is, does it work in 3D? So if you have shanks with electrodes at varying depths, or is it only 2D right now?
It is 2D because of the system that we work in, but I don't see in principle why it shouldn't work in 3D. So if you know anything about the machine intelligence techniques for doing feature analysis, it's primarily based on a self-organizing map. So I'm using the positional information that we're getting out of multi-electrode arrays, because we know exactly where each electrode is in reference to each other. I'm using that to triangulate neurons, but that should work in 3 dimensions as well. I could go to, right now I'm using a 2-dimensional self-organizing map. I could use a 3-dimensional self-organizing map, and it should work exactly the same.
And then I was just going to ask you to talk a little bit about the algorithm you're using to do spike sorting. Is it mostly based on the PCA?
No, I'm not. But that I could be using. It is self-organizing maps that I'm using to do spike sorting. So I'm doing triangulation and spike sorting in one step, which I think is my claim to novelty here. But I could make my job easier by doing additional feature reduction by using something like PCA or ICA. I haven't really explored them deeply yet.
Yeah, I had a question sort of about the nitty-gritty of data analysis that you mentioned. I mean, you mentioned using the Granger causality. Is that right?
That's correct.
Does that work well for you?
So we've had some problems with it. One of the big areas, so the next paper I'm moving on to after this is actually looking at the different functional connectivity measures for spiking data, for spike trains. I had some problems with it initially. Granger causality works best with smooth data. It was really developed in econometrics.
Yeah, I mean, I've tried to use that in other contexts. Then we had to develop other methods instead, because I often wouldn't find causal patterns that even you had put in there in test data. I think so. I was curious if this data was such that it would work well, but.
So a lot of it's probably going to depend on the neuron's perception of other neurons' spike streams. And so when you're trying to pick up this functional connectivity, we might need measures that actually do something similar to how the neuron is statistically processing.
Right, because I mean, in theory, Granger causality means you want to find some function that will produce the output given the input, and you don't know what kind of function you're looking for. So the typical version makes some assumption about that, and then.
Yeah, there are quite a few papers coming out this year now. There's I think 4 or 5 already on using Granger causality with spiking neuron data, and we'll probably be contributing to that slew of papers as well. But yeah, this is a big topic that's on the stage right now in neuroscience.
Yeah, I guess my question, just thinking as someone who does data analysis research, is like one line of research I pursued years ago was better ways of causal inference than the standard Granger causality methods, and I'm curious if that line of research is potentially interesting for the kind of work you're doing, or if it's sort of beside the point.
No, absolutely. It's very interesting. And, yeah, like I said, it's a very hot area in neuroscience right now.
Cool.
Yeah, we have tons of data streams that we need to look at. We want to find out where the information flow is between those data streams.
Yeah, but do you know the correct answer, and you can test your causal inference method, or?
So there's a couple ways we look at this. One thing I didn't mention that we work on, I'm already using simulated systems. So the validation system I use, I'll do a simulated neuron model. I like the Izakevich model. So I'll do a network of model neurons where we know the connectivity. And then
And so how well does Granger causality do it inferring the known causal relations in a network of Izakevich neurons?
Fairly well. So it does pretty good at that for small networks of neurons. And a couple other people have shown that already. There's a couple papers, if you're interested, I could send you. So for defined networks, you can pick it out because you can set the parameters for network discovery exactly as they need to be.
Okay, so you set the parameters of the algorithm based on what you know about the network, because you set the network up in the first place. Yeah. Sounds like a good way to publish papers. Yeah.
Yeah. So the key is getting the time lags correctly, because Granger causality, basically for it to work correctly, you need to know what lags you need to be looking at, and if you have to look over very long time periods, over very long time lags, it becomes very computationally intensive to do these techniques. So basically figuring out how to figure out the right model order is one of the critical questions for any of these functional connectivity measures.
Yeah, I'd be curious for you to point me to some of the better papers on this and see if it connects with other stuff I know. Thanks.
Is there another question, Peter? You have one?
Yeah, whole emulation.
Okay, why don't you ask the question?
So, do you think that it would be a useful objective in the quest for whole brain emulation to do a whole snail brain emulation? Would that be useful in your line of biology?
That's exactly what I'm working on.
Okay.
That's sort of the point of this.
Peter, I'll ask you a question myself. Do you think that right now, from the data that you're getting out of Limnea, that you're learning something perhaps about resolution, about the significance of types of signal we're looking at, and the kind of things we have to pick up?
Yeah, I mean, so one of the things I'm really interested in is how do we parameterize these models using very low-level data so that we can get higher resolution or higher level representations. So if I have a large set of neurons, do I need to have all of their single neuron voltage resolutions, neuron voltage records? Sorry, I'm getting an echo, so it's a bit confusing. So do I need the information from all the neurons in that system in order to get a full understanding of that system's dynamical space? Or are there's some neurons in there that I can just use probabilistically? Are they only modulating the activity in certain circumstances, and that I can just write a simple function that will represent the modulatory activity of that neuron, that I don't need its full voltage record? So those type of questions. How do we take low-level data and do this type of compression that you were talking about earlier? So it's those types of lessons that I think we're just beginning to get to now. The other thing I think we're getting to is, because we can do these learning and memory experiments in a fully observable system. So this feeding experiment that I was talking about, if we train the brain outside of the organism to respond to a particular taste, what actually happened in that network in response? So how did that memory of responding to the neutral taste, how did that get formed? And we can look at that at the network level. And this is only by using a technique where you look at lots and lots of neurons at once are you able to pick up this type of information.
Thanks, Peter. One more look to see if there are any other questions from the live. Yes, Michael Andrew, next question.
Hi, Peter. I was just wondering about the electrodes that you're using.
Yep.
Are you making those yourself? You're fabbing them, or?
No, these are commercially available. We're using a multi-channel systems. They're out of Reutlingen, Germany. A 252 electrode system from them right now. And I'm very interested in, there's a company called 3Brain that just came out with this 4096 commercially available system. There are some labs around the world that are fabbing their own, though.
Okay. So that's the highest number? That's like 64 by 64?
Yeah, so commercially available right now.
How long are they? How long are these like needles? I guess they're metal needles extending out of plane, or just a patch of metal.
Yeah, so it's essentially all of these systems that I'm talking about. There are glass petri dishes with planar electrodes embedded in the base of the glass petri dish. The techniques I'm talking about, the analysis techniques, could certainly be used for penetrating electrodes and things like that. I would just have to extend my methods to working with three-dimensional data.
Thanks.
20 microns seems just really big, though, patch of metal. Oh, hi, Peter. I was just wondering, like, 20 microns seems really big for like a patch of metal. It seems like for getting higher resolution, it should be really easy.
So I mean, we use electrodes that range from 10 to 30 microns. But so what was the point about that it's a big patch of metal?
It just seems you were mentioning that it would be beneficial to get higher resolution, or just more sensors per neuron. Yeah, it just seems like that's, I don't know. It doesn't seem like a very I mean, like computer chips or so. I mean, they're doing like 22 nanometer or something line widths. But
so some of the practical trade-offs we face. If you use a larger electrode, you get a much better signal-to-noise ratio. So yeah, if you go to a smaller electrode, you can get smaller feature sizes, and you can maybe get more sensors per neuron. But then if you go to a larger electrode, you get a much better signal-to-noise ratio. So this is the stuff we're dealing with. So we found recently these 252 electrode arrays, they're 30 micron electrodes spaced 100 microns apart, and you get very clean signal from lots of neurons, and you get very clean signal from lots of neurons on lots of electrodes. So that actually makes my job easier if I can pick up a neuron on more than one electrode, because I can triangulate it and I can estimate its size more easily by doing that. Rather than if we use a 10 micron electrode, the signal noise ratio might be worse. I'll only pick it up on one electrode, though, so you might be able to triangulate that way by just saying, okay, it's sitting on top of this electrode.
Okay, and I'm sorry, I just had one more quick question about just how these sensors work. Is this like an electrical signal, or is this like detecting ions of calcium or something?
No. So we're looking at extracellular voltage.
Okay.
Yeah. So you can do current source density analysis, which is another technique that I work with, and basically you reverse calculate what's actually happening inside the neuron based on its extracellular voltage.
Thanks. We have another question coming through, Peter.
Hi, it's Tal again. I just wanted to make one quick comment further about the analytics piece. I don't know if this will be useful or not, but we have a rules-driven, arbitrarily dimensional hierarchical modeling environment for organizing analytic problems, and we've used it in a bunch of different domains for exploring what sounds like it might be akin to the feature separation kinds of problems. A lot of times it's been used in sort of market analysis stuff where you have, billions of transactions going on and you want to figure out why people in Chicago like strawberry yogurt more than blackberry yogurt. But anyway, all that stuff is completely abstracted in this modeling environment that we have. And if it's useful, I'd be happy to help anybody who may be interested to understand whether it would be useful for them or not. But we'd be happy to make it available for research purposes to anybody who would want to play with it. I can't commit to making it open source standing here right this minute, but I can commit to providing it for non-commercial applications to anybody who might want it. We spent about $80 million building it. It's a pretty cool thing. Used it on some really massive problems before, but for what it's worth.
Yeah, I mean, I'm certainly interested in looking at any tools that will make my job easier. I'm recently getting very interested in. I'm a recent Python convert now. I've done a lot of my programming in low-level C, but there's a good place to look for resources on this stuff is the INCF, the International Neuroinformatics Computing Facility, to see what people are actually using to do these sorts of large-scale data analysis. There's a website I like quite a bit called Neural Ensemble. One of my big bugaboos with neuroscience is basically everybody comes in, does a PhD, they write their own software package, and that so there's a lot of information lost. We really need to start getting to these large-scale systems where everybody uses the same system and the same tools so that we can compare papers to each other, data analysis methods to each other. But yeah, so anyway, point being that I'm very interested in looking at other tools and other packages, because one of my big failings is I think I tend to want to write everything myself, and I'm trying to get away from that as much as possible.
Well, as a parting gratuitous comment, because I'm a major fan of the language, our thing has a nifty Python interface that you can drive it from.
Very cool.
Thanks.
Another question coming up.
I noticed in the discussion of the scale at which particular neural processes are occurring. This reminds me of kind of the question of, if you take a before and after, like people who do brain shape research, where they're working with like commercial apps that train your brain a certain way, and they do before and after scans to show how functional development changes just with a particular concept being driven in over and over and over again over time. Are you aware of any places I should look or answers about different categories of concepts or different categories of brain trainings, and exactly at a neural scale how widely these things change, and if repeated exposure is deepening the same neural connections, or if it's continuing to sort of build new connections all over the brain? From a mimetic perspective, if you were going to talk about how many neurons in a meme, if you will, it's like how many angels fit on the head of a pin, right?
Yeah, so I mean, I think the problem there is that we don't have the experimental systems to do that on the scale of vertebrate brains right now. Probably the only place to look for experimental data like that. So if you look at some of the work that's been done at Duke by Nicolelis's group and the Brown group, the cyberkinetics people, they're starting to do sensory experiments where you look at motor training and things like that, and you look at lots and lots of neurons at once, but really the data that you're looking for doesn't exist. And that's, we need to get to generating that type of data. We're just now starting to have the experimental systems to try and address those types of questions.
Thanks, Peter. I wonder if there are any more questions from the room. No, looks like that's. Oh. Okay, Luke Nosek wants to know, how does measurement with electrodes compare with measurement, say, with optogenetics, for example?
So you can't really measure anything with optogenetics right now.
Right.
Optogenetics
okay.
is
but let's just say bioluminescence.
Okay, yeah. So voltage sensitive dyes are a better technique to compare. So the signal noise ratio on voltage sensitive dyes has been particularly bad, but some people people are developing new dyes which seem to have much better resolution. If you wanted to look in invertebrates, there's Bill Frost. His stuff is getting very good now. So it's the big advantage with voltage sensitive dyes is you know, because you're looking visually, you know what neuron it came from. If you can visually separate the neurons out, you know exactly where that information came from. So you don't have a spike sorting problem. You can just look at the neuron and see that its signal is changing. But the signal to noise ratio has been a problem with these dyes, and they're particularly toxic. So these are sort of the experimental trade-offs. With multi-electrode arrays, the experiments are very easy. We can just basically slap a piece of tissue onto a planar multi-electrode array, and you're ready to go. So we have that advantage. But as I said in the talk, I'm very interested in using a combination of techniques if possible. So the other problem with the visual techniques is, well, yeah, I mean, this is the same thing. It's just an experimental difficulty because you need quite a bit of equipment to actually do the visual scanning with the voltage sensitive dye work, and you need to worry about perfusion and getting fluid flow over these systems, and so there's a lot to worry about in experimental setup there. And also I worry about with the visual techniques translating that to in vivo systems, where we know with electrodes that we can go in vivo if we want to. We can do an implanted electrode array. Doing an implanted system to do something like bioluminescence scanning, there's not a really clear technological pathway for doing that sort of thing right now. And I mean, one of the practical applications I'm interested with my stuff is eventually moving into neural implants, brain-machine interfaces. So this, for me, this is, I would prefer to use the electrode pathway because there is a clear technological path for getting from where I am right now to brain-machine interfaces.
Peter, then I'd like to add a question to that. So you obviously see this as research that leads towards whole brain emulation. And you were just mentioning that methods such as, basically using dyes and things like that seem to have a lot of complication. At least right now they do.
Yeah.
But how would you, like, you're saying, okay, for brain-machine interfaces, yes, it's clear how you could use neural interfaces based on electrodes. That's clear. But how would you move from this technology to something where you would want to be able to acquire data from a whole brain? Like,
yeah,
let's say
so,
a big brain, not just limnea, but something else.
Yeah. So, I mean, the sort of technological pathway I see myself following in my career is working with these invertebrate systems as a testbed to generate ideas about data analysis and brain-machine interface for interfacing to neural systems, then moving into brain-machine interface for very specific parts of neural systems. I'm sort of counting on, for the large-scale stuff, for eventually getting to interfacing to entire brains. I'm counting on nanotechnology or future technological developments for doing that type of work. I'm not, basically anything I think any of us say right now has to be taken with a grain of salt, because we don't know where the technological development pathway is going to go. But I do think that this is the clear technological pathway, is using some sort of implantable sensor to get out most of the information that you need. And yeah, this is all speculation how we go to whole brain emulation in terms of what information we need. But I think the type of systems that I'm working with, from the bottom up perspective, you'll start to generate these model systems that can tell you how good your particular information is about modeling a given system, and what information you actually need, which is, I think, the critical question that none of us really can answer right now.
Right. And so to just summarize, you would say that your approach is very useful for understanding what types of information we need to look for in a neural system, and also for brain-machine interfaces, but that you would look for other technology, other tools to go beyond that. Yeah, so I think that what I'm actually up to is developing a general rationale for how you attack these systems. And it's, I'm trying to be sensor agnostic. Obviously, I'm focused on the tools that I have right now and developing analysis methods that work with the tools I have right now, but I'm also very much keeping an eye to making them as general as possible. That, I could be looking at time series, at voltage streams, coming from voltage sensor diodes or from multielectrodes, or fMRI voxel data or EEG records. It's all just time series data, and looking at the connectivity between that time series data and seeing how dynamical patterns emerge from either neurons or parts of brains that are connected to each other. Thanks, that's great. I don't see another question from the room right now. So, online question at this point? Sorry, yes, Peter? I was just going to say one other comment before we move on. So I had mentioned it in text chat earlier, but I really don't want to see divergence between AGI and neuroscience. I think that's one of the large problems we're facing. Sort of my shtick for my career right now is application of machine intelligence to large-scale data sets in neuroscience. But I think there's a lot in neuroscience that can inform AGI as well. I think that there's a lot to be learned from neuromorphic engineering, from what these brain systems are doing in terms of manipulating information in a dynamical system sense that you could apply to AGI systems as well. So I'm very interested in getting more crosstalk going between artificial intelligence and neuroscience, and I still don't think there's enough of it going on. Mm-hmm. Yeah, I tend to agree with you personally. I think that Ben Gertzl might not agree. I just woke him up, I think. So. Ben agrees that I just woke him up, but he may not agree with your point. Peter was just. Tell Ben I have no sympathy for him. It's coming up to 6 a.m. here. So basically, Peter was just trying to inform us that we shouldn't give up on having a connection between neuroscience and AGI and whole brain emulation and all these different areas, because neuroscience can inform the development of AGI, and AGI, or AI, at least machine learning, can help in the development of tools, for example, for whole brain emulation. I totally agree that it's machine learning development. Question I was asking about Granger. We did some work in the late 90s using genetic programming to make improved versions of Granger causality, the causal patterns between the data sets. We applied this to financial data sets. Definitely use machine learning for causal inference.
Could the speakers come to the mic, please?
Yeah, I was just going to say that Ben says you're full of it, but I think he wants to tell you. You tell me.
You're very full of it, yeah. No. I mean, I think you can definitely use machine learning in all sorts of powerful ways to help with this kind of data analysis. And what I was getting at before, I was thinking of work that we did in the late 90s using genetic programming to improve on typical Granger causality, to infer causal relations between financial time series and biological time series and so forth. So, I mean, that's definitely a big role for machine learning in helping with causal inference, and then with network inference and network analysis and so forth. Whether you need general intelligence as opposed to machine learning tools to help with this kind of data analysis is kind of an open question. I'm not sure, and I'm more sure that you do for genetic data analysis and longevity research than for this stuff, but I haven't done as much work in this kind of biology either. So where you draw the line between needing advanced machine learning and needing an artificial biologist to help you crunch the data. But the other way around, like what use is the whole brain emulation work for AGI? I'm more skeptical of it, partly because my approach to AGI is not based on spiking neural networks or anything that's closely brain-like. But then it all comes down to what level are you studying the biological system. I mean, the molecular level pretty much can't be relevant to building AGI systems, but maybe at a much higher level it can be.
So what I'm speaking of specifically is, I mean, the machine intelligence techniques I've selected, I'm working with things like self-organizing maps. So both in terms of self-organizing maps and Granger causality, the brain is doing feature selection in ways that we don't completely understand, and doing it very, very efficiently. So I think there's a lot of, I'm interested in developing these sort of cyclical systems. So if we have a machine intelligence system that can analyze neural data and can understand the type of feature separation that the neural system is doing, and the type of causal relationship processing that neural systems are doing, and learns to emulate that, sort of forming a virtuous cycle of, But
I wonder if the human brain is actually well attuned to understand low-level data about the human brain. I mean, maybe it isn't. Like, finding causal relations between neurons is quite unlike the causal relations that a primitive dude needs to find to get sex and food in the jungle. So I don't know if the virtuous cycle really exists. Maybe these data analysis problems are quite different than what the organized activity of the brain is meant to do.
Well, I think I mean, this is just personal speculation, but I think what's been going on in primate evolution is that the cortex has been getting better and better at generic problem solving. And so I think we'd like to go in and see what's going on in the dynamics for how it's actually done that. If you look at sort of
I guess I spent too much time as a university professor to think the brain is good at generic problem solving. I mean, so my intuition is a little different. Look, I feel like the brain's holistic activity is tuned and biased to recognize some kinds of causal patterns in networks, whereas the ones that occur in sort of lower-level neuroscience data analysis may be quite different than the ones the brain is tuned and biased for. Not that we can't solve those problems. Obviously, we can, and we are, and you will. But maybe the kind of biased general intelligence that the human brain is very far from the optimal kind of biased general intelligence to puzzle through neuroscience data. And that would be my own guess, which is also a speculation.
I guess just final comment on that is, yeah, I feel your resistance, and it's sort of frustrating to me that there has been quite a bit of resistance to neuroscience data and among people working in artificial intelligence. But I still, and my bias as well, coming at it from the perspective.
Well, I mean, I don't know if there's a resistance to neuroscience data. It just hasn't been useful, and you read the papers, and it doesn't help. Like there's no problem for which a brain simulation works better than the best of breed AI algorithm for that same problem yet.
Well, well, not an emulation, but certainly humans are better right now at AGI than AGI is, right?
Yeah, well, certainly, but there's no case where looking at the brain has been the way to get the best solution to some practical or computational problem yet.
So, I mean, that's sort of my point, is the problem you're trying to solve, the human brain is the proof of concept. So if that's the problem you're trying to solve, you should be looking
Oh, but it's not the problem. I'm not personally trying to create a digital emulation of a human brain. I'm trying to create an intelligent system.
No, you're trying to create, a general intelligent system, and the only existence proof, all I'm saying is the only existence proof we have right now is the human brain.
Yeah. Well, there are other animals, but I mean, I think that there's a question here of the dual goals, which are the different goals of trying to emulate the brain and trying to understand how the brain works, and you're kind of sort of trying to do them both at the same time. And of course, one could also separate them and try to understand the brain on a higher level in a way that wouldn't be much use for replicating a particular guide digitally, or you could try to emulate the brain in silico in some way that would be very little help to understand the brain. So it's interesting that you're doing the two of them together, which not everyone is anyway.
Yeah.
Or trying to do the two of them together.
There was a comment I caught just a bit of earlier about you could do a really high resolution scan. You could do a molecular scan, and just if you had a decent molecular simulator, and emulate a brain, and you wouldn't have any clue about how that was actually working if the molecular emulation was working really well. You wouldn't have any idea about the layers from molecular on up, all the types of data processing, this sort of different layers of data reduction things going on. You would have no clue. Similarly, you can go to the very high level, and if you were successful in building an AGI system, you could build something that only represented the highest dynamical features that we might see in the human brain. So the point I'm getting at too is that I think the easiest way to do this is to go at all levels simultaneously, so you understand the system from top to bottom.
Okay, thanks, Peter.
Yeah.
Yeah. So thank you very much, and also for the debate, you two. We have to start closing this, so that I'm going to try to make a couple of points towards the end here. One of them that I wanted to make is that I think that we still don't have that much insight right now into how we would advance the problem of, resolution, let's just say, although I think that Peter's approach is definitely something that's feasible in small brains to understand more about what kind of signals we need. And perhaps it is, in fact, a matter of trying to have test systems that we can learn from. So this Limnea is exactly one of those examples. We've sort of looked at the me program issue, and it looks like it may be potentially useful if you're looking at something with maybe less ambitious requirements or less ambitious goals as to what you see as a re-implementation of functions of the mind, but may not be such a great help for whole brain emulation or advancing substrate-independent minds at that level, except by validation, as a validation model. But finally, what I really wanted to say as a last point, or actually ask you all, is what would you like us to do next? What would you like us to do so that you can see progress when we next have a meeting, perhaps after we solve a lot of technical problems that we had this time? But besides those technical problems, what else would you like us to do? Which kinds of, let's say, I don't know, literature or advances through certain projects, or what sort of steps do you think would be necessary to make sure that we have progress in ASIM? Sorry.
From the point of view of people working on data analysis, it'd be nice to have like standardized test data sets which are relevant to whole brain emulation, where you could try out causal inference or network inference algorithms and see if your algorithm is doing better at kind of incrementally working towards whole brain emulation than the other algorithms. Maybe these exist very thoroughly, and I just don't know about them because it's not my area. I don't know.
And they are now. People are starting to assemble these test data sets.
Is there some web page that might be developed data sets for whole brain emulation?
Sorry, I couldn't hear that.
Is there some like central resource for this, like somewhere I could go and say here are the standard test problems for analysis algorithms useful for whole brain emulation or network inference or something?
Yeah, I mean, there's a few different places. The INCF would be one clearinghouse. I forget the U.S. site now, but where people are basically they're developing data sets from particular experiments, storage areas for data sets from particular experiments. So one example is the Kokomo database is a good example.
Yeah, I know that one. Right.
Yeah. So these types of databases are starting to come into existence now. But I could certainly assemble a list of them. Like I said, the International Neuroinformatics Coordinating Facility has a fairly decent website that lists a lot of these different resources.
Cool.
I was rushing ahead a little bit based on the time being 10 o'clock, but we'll just let them throw us out again. We actually have one more talk coming up right now, though I'm not sure how it's showing up in